Welcome to this, our second Ask the Expert SSERC meet. The topic today is Understanding the Genome, and our expert is Professor Jerry Graham from the Institute of Infection, Immunity and Inflammation at the University of Glasgow. Jerry has long been a friend of SSERC and a supporter of and a contributor to SSERC events. This means that he is also a great supporter of biology teachers and biology education in Scotland, and it is my great pleasure to welcome him.
So over the next half an hour or so, what I want to try to do is explain to you some of the science behind the genome, how the genome was sequenced, what it's told us about biology, and actually very importantly what it's told us about disease, how we deal with disease, and actually how we respond to therapy. So there's some important messages to come out of this. So the genome is a remarkable biological achievement. What we managed to do is we can squeeze 1.5 meters worth of DNA, 3 billion bases worth of information into the nucleus, which is approximately the size of about 1/100th of a millimeter in diameter. So the entire information required to recreate me as a human being is packed into the nucleus of every single one of our cells. It's a tremendous information content. And so if you want to understand a human being, if you want to understand any species really, then looking at the genome is essential because it's basically the blueprint for life. And what's also become extremely clear is that it's not just life, but it's death is imprinted into the genome as well. And so many diseases are written into the genes, our responses to therapy written into genes, but also our prognosis written into genes. So all the genome information helps us understand biology, but also helps us understand disease and how we respond to disease, and we'll try to look at these as we go through it. So the fact that this is the blueprint for life, it's a key player in disease, triggered people some years ago to decide that they wanted to read the sequence of the human genome, to read the 3 billion base pairs of the so-called blueprint for life. And this was called the Human Genome Mapping Project, and it was started, as I say, about 25 years ago, substantially funded by the UK and US governments, although later on I'll talk to you about another aspect of it, which was commercially funded. And the major aim of it was to read the entire sequence of the human genome. Now that's really quite a challenging prospect because trying to read 3 billion bases across all the different chromosomes we have represented a major technical challenge. So a lot of the genome mapping project required development of really completely novel and extremely complex technologies, fundamental of which was the ability to read a sequence of that length. The original sequencing methodology that was used, sorry, I'm struggling. It was Sanger sequencing. So Sanger sequencing is one of the traditional methods for sequencing, and it involves what's called chain termination. I'll explain that in a little bit more detail, but essentially the reaction involves taking a template, which is the genomic DNA. I'm just trying to get my cursor here, the genomic DNA, and little primers that allow you to prime a reaction that copies that template. And to help copy the template, we have the deoxynucleotides here, which will be incorporated into the growing chain. And what we also have in the reaction are coloured nucleotides, which are so-called dideoxynucleotides. And these are important because these are different from the deoxynucleotides because they contain in them, instead of a hydroxyl group, they contain a hydrogen group. And what that means is you cannot extend the chain beyond them. So once it's incorporated, the chain is terminated. And this gives rise to the name of this sequencing methodology, which is called di-terminator sequencing. So essentially what happens is that the chain is copied, but it randomly inserts these di-terminator fluorescently tagged nucleotides and stops the chain. And so you start to be able to stop at different points, and one can then read the sequence. So you can read the sequence by running these fragments out on a gel. The gel is read using a laser, and it's usually reported in this sort of way here. So this is di-terminator sequencing. It's essentially what's called Sanger sequencing. It's one of the things that got Sanger one of his 2 Nobel Prizes, which is a pretty impressive achievement. And one can read approximately 1,000 base pairs, or 1 kilobase, using this methodology. Now there are much more contemporary technologies. I've just disappeared off the screen. Is that okay? I'm back, sorry. So-called next generation sequencing methods, which we don't have time to go into because they're pretty complicated. but they involve what's called massively parallel sequencing. That is, they sequence lots and lots of different fragments at the same time. In a second, you'll see why that's important. So the basic genome sequence involved Sanger sequencing, specifically this thing called di-terminator sequencing. But we still have a challenge, and that is that if we can read only 1,000 bases at a time, how do we knit all this together to give the entire 3 billion base pair sequence of the human genome? So what we have here is about 500 nucleotides long, and our genome has about 6,000 times that length. So how do we realize that sequence? And the way that we do it is we take lots and lots of smaller sequences and we stitch them together using computing. We're going to talk about that in a minute. And the method that's used is called tiling. And tiling is quite an easy method to understand. And let me give you a relatively simple analogy to explain how tiling works. Here's 4 parts of a page of text, and they've been cut out. If you want to knit them together to find out what the page actually looks like, you look for overlapping pieces. There's pretentious Latin style, there it is there. There's metaphors there, and there's, I can't actually see here, but anyway, one can see the overlap here. So by finding the overlaps, you can knit them together and you can generate the original page that it came from. And that's a simple analogy, but that's really essentially how the genome sequence was put together. It involved massively parallel sequencing. That means lots and lots of small sequences. It says here up to about 430 base pairs, but it's actually up to 1,000. Covering the whole length of the genome were put together, and then the overlaps taken into account, and then you can, from that, you can infer the entire sequence of the genome. Now, this is not a trivial undertaking. This is not something you can just do in an office. It requires extremely complicated mathematics and statistics. And this really gave rise to a brand new type of biology or biological science, which is called bioinformatics. And bioinformatics we define as an interdisciplinary field combining computer science, statistics, mathematics, and engineering to study biological data and processes. So essentially what bioinformaticians try to do is to take extremely complicated biological data sets that you cannot reasonably deal with on your own and distill them down in a relatively simple way using advanced computing and mathematics. And so we'll come back to bioinformatics a little later on, but this is an extremely important advance. In fact, it's no exaggeration to say that bioinformatics and the computing expertise of bioinformatics has actually driven much of the success of the Human Genome Mapping Project and some of the other things that I will show you a little later on. So there's a rule in computing, and it's called Moore's Law. And what Moore's Law states is that, broadly speaking, the processing power of computers doubles every 2 years. And this green line here that you can see, let's get the cursor again, this green line here roughly follows the Genome Mapping Project. And you can see that it follows Moore's Law, but then it accelerates way beyond it as bioinformatics took off as a speciality and developed some extremely complex computing tools that help with this process. And as I say, it is no exaggeration to claim that bioinformatics has really been responsible for driving the success of the Genome Project and a lot of things that are downstream of it, some of which we'll talk about a little later on. So to put the success of bioinformatics in perspective, the original genome sequence, which was reported in 2001, took 13 years to complete and cost $3 billion, but now, in 2014, we can sequence a genome overnight. Not sure what happened there. That shouldn't have come up. Sorry, something's gone full screen, which I must have clicked something. Thank you. So in 2014, we can sequence the entire human genome overnight. So we've gone from taking 13 years to being able to do it overnight. And it doesn't cost $3 billion. It's actually less than $1,000 to sequence an entire human genome now. And as I say, much of this advance has been driven by bioinformatics, which is a special, new and extremely potent and important branch of biological science. So the genome is, broadly speaking, sequenced, and the question then is, what does it look like? Well, there are a number of surprises in the genome, and I'm going to tell you what the genome looked like 10 years ago or 15 years ago when it was sequenced, and then we'll look at how our understanding of the genome has evolved over the intervening period. So in 2001, when the genome was sequenced, there were two major surprises for people. One is that people assumed that we would probably require around about 250,000 genes to make a human being. In fact, we're only about 10% as complex as that. We only require around 23,000 genes, and that was really something of a surprise. The other surprise, and we'll come back to deal with this point specifically in a few minutes, is that the bulk of the genome appeared to be junk. Only 3% of the genome actually encoded for genes. The rest of it, 97%, had no apparent function, but we will refine this issue a little later on. So what does the genome actually look like? The genome is, as I say, a government-funded initiative. You can go online and look at the genome. It's not a great read, it has to be said, but an analogy that I like to use is the analogy of using a dictionary. And it's like reading a dictionary but having pages and pages of random letters, reading into a word and a perfect definition, more pages of random letters, another word and a definition, etc. So the idea of the genome initially was long stretches of random bases, but little pockets of beautifully described and maintained information, which of course we call genes, interspersed with more long stretches of random bases. So the idea of the genome as a dictionary is actually a good one, because we also use the terminology or language to explain really pretty much everything we do with the genome. So we transcribe it into RNA and we translate it into proteins. And we're not going to talk about this at all because it's a separate talk in its own right. But although I say we are about 10% less complex genetically than we thought we were, we are wildly more complex in terms of proteins than we thought we were. And there's an entire branch of science around proteins, which is called proteomics, which starts to get to grips with the really remarkable complexity of proteins that we didn't realize that we had. So that's roughly what the genome looked like. And the 10th anniversary of the genome sequencing was celebrated in 2011. We're now 4 years on from that. And in the intervening period between 2001 and now, there have been very many advances in understanding the genome that have really, frankly, changed a lot of the ways that we understand biology and changed the ways that we appreciate how the genome functions. So, as I said, initially, we had this idea that we have 23,000 genes, which is still pretty much the case, but they only occupy 3% of the genome. Now, that is broadly speaking true, but it turns out that much more of the genome is actually given over to some kind of function than we initially suspected. So, for example, and I'm sure many of you know this, I've cheated slightly here, so I represent genes here as single blocks of information. And of course they're not. They're split up into exons and introns. And what we've seen from the genome information is that these exonic and intronic structures are spread over much longer distances than we initially thought, and we found exons for genes that are far distant from the original gene that we didn't expect. So we are now aware of the fact that genes extend over much larger distances than we initially thought. The other thing that we've become aware of is that really quite a substantial part of the genome is given over to regulating the expression of genes. So genes have to be regulated in an extremely subtle way in terms of time and space. And I'll give you two examples of that. One is globin. So I breathe through red blood cells and globin. So I make globin in red blood cells. I don't make globin in my brain or my skin, etc. So something tells globin to be switched on in red blood cells. Something also tells globin to be made at higher levels if I go and live high up in the Alps because I need to make more of it. So the subtle regulation of gene expression. Another one is the gene for actin. Actin is a protein which I'm sure you all know gives the basic structure to all cells. So it's present in all cells. It's like the cytoskeleton or the scaffold of cells. But muscles, of course, muscle function is dependent on actin. Actin is a major part of the machinery that drives muscles. So muscle cells have to make a lot more actin. You have to therefore switch the gene on more effectively there. And if you're exercising, you make more of it. So again, it's regulation of gene expression in time and space. And put relatively simply, gene expression is regulated by so-called promoter regions. And these promoter regions are regions of non-coding DNA that sit at the left-hand side or 5' side of the gene. And these are, if you like battery packs. When they're empty, you don't get transcription. But when so-called transcription factors, or very often transcription factor complexes, bind. That then triggers transcription, and you can drive transcription of the gene. And this can be extremely subtly regulated in time and space, and as we'll see later on, actually accounts for a large part of what it is to be human and how we distinguish ourselves from other genetically very closely related mammals. And as well as promoters, we have enhancer elements and lots of other regulatory elements that occupy a large part of the genome. Now, the other thing, I'm just fishing around for a hanky here, my apologies, that's my hay fever kicking. Genome that we simply did not know existed 10 years ago, but are actually of profound importance. I'm going to talk to you about these in the next slide in a bit more detail, but there are things called microRNAs in the genome. We have around 1,200 of these. As the name suggests, these are very small genes that encode extremely small RNA. We have other issues. We have these long non-coding RNAs. We have chromatin structure, so the way that chromatin folds actually regulates gene expression. The genome is full of viral sequences that regulate our own gene expression, strangely, and there are lots of other things to be learned as well. So the genome is much more complicated than we thought. And I want to just spend 1 minute talking about these microRNAs, because these are new, they're in the genome. As I say, there are around 1,200 of them, but they are of really profound importance. And I don't want to go into this diagram in any detail, but what microRNAs do is they attach themselves to the end of messenger RNAs and they lead to their degradation. So they help regulate transcript levels. And I want to show you just one of these. This is called microRNA 146. This is the entire sequence of it. It's only 22 nucleotides long. It's what we call a 22-mer. That's all it is. But if you take those 22 nucleotides and you remove them from the mouse genome, you generate mice that are profoundly sick. So in red here we have wild-type mice, and these are mice responding to a relatively innocuous inflammatory stimulus. It really doesn't trouble them, but the knockouts have major problems. They simply cannot regulate inflammation. And even without inflammation, these mice in which you don't have the microRNA have massively larger spleens, which suggests something's going wrong with the immune system. These are lymph nodes, which again are much larger in the mice that don't have this microRNA compared to the normal ones, so something's going wrong here. And these figures we don't have time to go into, but essentially they show that these mice also spontaneously develop a range of cancers. So these microRNAs are new, they're of profound importance, they're tiny, but getting rid of them has major consequences for mice and for us. So the genome is now defined. We have a lot more information about what's going on in terms of other things in the genome than we thought. And about 10 years ago, there was a consortium called ENCODE, which is an encyclopedia of DNA elements was started. This was started by the bioinformaticians, these computer specialists who can analyze the genome, and they hooked up with biochemists, and what they tried to do was to analyze all the genome sequences to look to see how much of it has some kind of function associated with it, to try to move away from this idea that 97% is junk and see if there's function within that 97%. And it was spurred on by the fact that 3% of our genome is genes, as I said, 1% encodes for protein. But so-called single nucleotide polymorphisms, we'll return to these later on. These are variations in gene sequence that are conserved within populations. A lot of these, in fact the vast majority, sit outside of protein coding regions, in parts of the DNA that we thought were junk. That suggests, because these are conserved, that they're doing something. And so what ENCODE wanted to do was to work out what was going on here. And so they went through the genome, they looked at all the coding sequence, they mapped all the promoters, every regulatory sequence that they could, they studied chromatin structure, histone modification, and the microRNAs and other non-coding RNAs that I mentioned to you. And they came up with quite a startling conclusion, and that was that, in fact, it's not just 3% of the genome that's important. Up to 80% of the genome they claimed was functional. So this work was published about 2 or 3 years ago. to much publicity because it suggested that we had grossly underestimated how important aspects of the genome were. But in fact, it's been extremely controversial, and there's been big discussions. It's a slightly geeky point about what is function, and what is function in the genome, and what is junk, and what is garbage is a distinction that's been brought out. And they actually got themselves into quite a lot of trouble. And what's probably a reasonable figure is that taking account into everything, into all these different species that we've got here, promoter regions, regulator regions, etc., approximately 20% of the genome probably has some kind of function. The rest is junk in the sense that it's perhaps waiting for some kind of function to be imposed on it, but it's not clear that it's doing anything at the moment. So we have the genome. We have now really radically developed our understanding of the genome. We're still trying to understand what it's telling us. There are surprises like microRNAs coming out all the time. There's many to be found. But you now enter what's called the post-genome era. And in this post-genome era, with the massive acceleration in our ability to sequence DNA, driven, as I say, in large part by the success of bioinformatics, we've gone from just having the human genome to having probably in the region of 6,000 different genomes available to us. Many viral pathogen genomes, this is the human immunodeficiency viral genome, genome of other mammals, the rat and the dog, genome of other vertebrates like the chicken, and genomes that are of agricultural importance. And here's a couple of other ones, the malarial parasite and the potato blight. And so the genome technology has moved beyond understanding human biology to understanding biology in general and actually contributing to things like agriculture and various other aspects of relevance to the economy. So it's now generally very broadly used technology. And we also talk now of entering the era of deep sequencing. And what we mean by that is the point I made earlier, that we can basically, with the advances in sequencing and bioinformatics, sequence any genome we want extremely quickly, a human genome overnight, and that's a major achievement. And that allows us to do a number of things. One is we can now routinely sequence pathogen genomes. So this is a mosquito here, the carrier of the malarial parasite, the Plasmodium parasite. And what you find in areas where malaria is endemic is that whilst drugs work up to a point, quite often the parasite will evolve in ways that makes it resistant to the drug. And that will be driven by its genetic sequence. So whereas previously what we had to do was to start to generate new drugs, what we can do now overnight is resequence the genome of the pathogen, understand what it's mutated to try to get it to become resistant to drugs, and then tailor the drugs to deal with that mutation. So the ability to rapidly sequence pathogen genomes has meant that we can play a really advanced molecular game of cat and mouse with genome sequences and help the design of novel drugs against them. Now, the second thing is that because we know all the genes, we can now rapidly identify disease-causing genes. And I want to give you one example of that that you may have heard of. There's a leukemia called chronic myeloid leukemia. And chronic myeloid leukemia is intensely studied because it's one of the classic paradigms for a so-called chromosomal translocation. So in chronic myeloid leukemia, 2 chromosomes break, chromosome 9 breaks and chromosome 22 breaks, and they misform. So a bit of 22 sticks onto 9, and a bit of 9 sticks onto 22. And just by an absolute freak occurrence, at this point where a bit of 9 sticks onto 22, you generate a fusion gene, and that reads through to give a fusion protein. It's called BCR-ABL for those of you that have heard of it. But the point is that fusion gene actually is an oncogene. And it's that gene that can kill you. And chronic myeloid leukemia was until recently inevitably fatal. But the point I want to make is that people could look down microscopes 60 years ago and could see this chromosomal translocation. But to go from that to identifying the genes that sit here and ultimately kill the patients took about 50 years. We could do it in a day or two now. That's the speed at which we can amplify the identification of disease-causing genes. And there's now a very efficient drug targeting this so-called fusion gene, which is called Gleevec, which you may have heard of. And that's a major success story. One still has to generate the drugs, but identifying genes causing disease, we can now do at an extremely rapid rate. So the other thing that we can do with the genome sequence is what's called data mining. And data mining is a new way of identifying novel genes. I'm not sure if any of you did molecular biology projects at any point at university, but before the genome was available, gene cloning was extremely laborious, and you would have an entire PhD project or a postdoctoral research project dedicated to cloning a single gene. What we have now are all the gene sequences known. We know every gene sequence. And so what we can do now, instead of having to clone genes by the old-fashioned method, is we do what we call in silico cloning. We clone essentially using a computer. And the way that we do data mining is if we are interested in a particular family of genes, I hope you can see this on your screen, it's a bit small here. Family of genes, of course, have conserved elements in them. That's what makes them gene families. And these elements are conserved because they're essential to the function of the genes. It's a family of kinase genes. This is a family of genes that I have a particular interest in called chemokines, which you've probably never heard of, but you see these cysteine residues are conserved. And what the bioinformaticians can do is they can write algorithms that allow you to search through the entire genome for conserved motifs. So essentially you can go through the entire genome in silico on a computer, and you can identify all the gene members of a defined gene family. That allows you to rapidly identify genes, allows you to identify novel drug targets, in fact major drivers of data mining with the pharmaceutical and biotechnology industries, and also identify disease-causing genes. Interestingly, and this started in about the mid-1990s, massive amounts of money were made from data mining. The reason being that as well as government-funded initiatives to sequence the human genome, there was a company called Celera set up, and Celera was a company that tried to beat the US and UK government initiatives to the genome sequence. And what they did was, as they sequenced the genome, they released it to biotechnology companies for a huge cost so they could do exactly this, that they could data mine to look for new kinase genes or new therapeutic targets. And so they made a lot of money. A lot of people cloned novel genes of therapeutic importance. They patented them. There are big issues surrounding that. But a lot of money was made, but perhaps more importantly, many novel drug targets came out of this. And in the mid-1990s, there was an explosion of gene discovery, and we now pretty much know all the members of the key gene families that we are. We don't know what they do, but we know what they look like in terms of gene sequences. So the genome has a number of other interests as well and values. One is in understanding the process of evolution. I'll deal with this relatively quickly. As part of the Human Genome Mapping Project, sequences of many other species were also gathered. One of the first sequences identified was the yeast genome sequence, of course, because yeast is the first eukaryote to appear on Earth. And there are many others: zebrafish, key experimental model, Drosophila, Caenorhabditis elegans, etc. The whole point of this, of course, was to try to understand the functioning of the human genome. But what it also allows us to do is to understand aspects of evolution, how we evolved as the supposed pinnacle of vertebrate evolution. And one of the other surprises evolutionarily is that in terms of mammals, we are remarkably similar to lesser mammals. So we're 98% similar to chimps, we're 92% identical to the mouse, we're not that similar to the fruit fly, but we're very similar to other mammals. So what makes us different? And it's not the fact that we have a larger genome. In fact, there's a bit of a misunderstanding about genome size. Mammals have a fairly tight genome size. Our genome is broadly the same size as the mouse or the chimp or other mammals. And in fact, some of the largest genomes belong to plants or some amphibians. So genome size does not relate to genetic sophistication or complexity. Actually, the key thing that makes a difference, that sets us aside from chimps and mice, despite the fact that we display very, very large amounts of homology with them genetically, is the way that you regulate gene expression. So I discussed promoters with you earlier, these regions that regulate gene expression. And it's these regions that are changed in evolution. They're major drivers of evolution. And they are major drivers of evolution because they alter the expression of genes. So alterations in gene regulation are major drivers of evolution. And we can start to understand this process because we have genome sequences from throughout the evolutionary process. And we can carry out what's called phylogenetic shadowing. And phylogenetic shadowing just allows you to line up sequences that are conserved through evolution. So here, for example, and this is these are promoter sequences. We'll just go through this very quickly. From early vertebrates right through to mammals. This little black arrow here is the start point of transcription. This little blue arrow is what's called a Tata box. That's conserved right through evolution because it's essential for starting transcription. But you can see while some of these elements in green are conserved all the way through these species, some are not. These elements here are only present in the mammals, suggesting some role of adapting this gene to mammalian function. So we can start to track the evolution of genes and how they're expressed using this process of phylogenetic shadowing. Incidentally, if you wanted to work out what this element here does, this little promoter element, it incidentally doesn't matter. It doesn't matter to this element what it has hooked up to it. It will drive whatever gene you put next to it. And so what you can do is you can take this promoter element, you can hook it up to a fluorescent marker gene, you can make a mouse with this in it, and you can generate what we call reporter mice. And so one can take these elements and understand where they're functioning by looking at the mice as to where they're expressed. So this one drives red expression up and down the spine of the animal. And what you also get are these beautiful images. This is kind of small here, but on a larger scale, this is a beautiful image of lots of different colored neurons, because promoters that drive different aspects of expression, different neurons, can be given colours. And these are called rainbow mice because it's a rainbow colouring within the brain. It's really very beautiful, but it helps us understand biology. So why is this important? It helps us understand evolution. It understands how we evolve genes and manipulate their function in mammals and how we make it more sophisticated as we develop. It also helps us to understand how the biology of these genes has evolved. So finally, I'd like to spend just a couple of minutes talking about personalised medicine. Personalised medicine is a major initiative that's come out of the Genome Sequencing Project. And the basic idea behind personalised medicine is this. Nowadays, in most cases, when we go to the GP or whoever we visit with a particular disease, we're treated in the same way that everybody else is. But of course, that's not necessarily the best thing. And the idea behind personalised medicine is the right drug for the right patient at the right time, because we don't all respond to drugs in the same way. Some of us will respond very well, some of us will not, and knowing that is extremely important. And the ability to respond to drugs is written into the genome, and the differences in our genomic sequences are predictive of this. Now, there are relatively simple personalised medicine initiatives going on anyway. There are mutations in the epidermal growth factor receptor in certain cancers, and the HER2 mutation of breast cancer. We know about these, and I think there are some case studies you'll get later on relating to these. But the genome allows this to be done in a much more sophisticated manner, and it relies on a number of things. One is identification of what are called single nucleotide polymorphisms, or SNPs, which you may or may not have heard of. And SNPs are single base changes in genes or in non-gene regions throughout the genome, and importantly, these are not random changes. These are seen in at least 1% of the population, so they're conserved for some reason, and they're usually biallelic. That is, it's just a single change. You don't get random changes here. So these are conserved, these are not random changes. We have over 1 million of these in the genome. There's one per 2,000 to 3,000 bases. They're mainly found in non-coding regions. If one looks at the distribution of them, 99% of them are non-coding regions. Only about 1 or 2% are in coding regions. And where they sit in coding regions, they can be described as being either synonymous or non-synonymous. And a synonymous change doesn't alter the amino acid, and a non-synonymous change does alter the amino acid. I'm going to show you an example of a non-synonymous one. It's a very famous one. It's been known for a long time. This is a beta-globin sequence. Normally has a so-called glutamate, which is a charged amino acid here, and a single nucleotide polymorphism here changes an A to a T and changes the glutamate to a valine. This is not charged. This is a non-synonymous change. This gives rise to sickle cell anemia, and of course is very highly studied, but it's maintained because it's actually of selective advantage in the populations amongst which you find it. So why are single nucleotide polymorphisms important? They're important because they can predict disease susceptibility and response to therapy. And what we can also do is we can use, I'm sorry, I've put this on, I've done something wrong there. sorry, my apologies. What we can do is we can use so-called arrays. These are little plastic so-called gene chips that have all the single nucleotide polymorphisms in that window. And we can measure all the polymorphisms in my genome or your genome, and we can use that to predict response to therapy, prognosis, susceptibility to disease, and we can do that at birth. So the other thing that we have are so-called genome-wide association studies. Most genes are polygenic, and trying to work out the genetic basis for diseases in which many genes are involved is extremely difficult, and one can't simply use the old family trees that we used to use. And so what we use are these single nucleotide polymorphisms, and you can define so-called allele frequency in the polymorphisms by taking these little chips. So what they allow us to do is to identify single nucleotide polymorphisms that are under or overexpressed in the disease population. Now that doesn't mean that the polymorphisms are actually involved in the disease, but what it means is that they probably sit close to a gene that's involved in disease because they segregate in meiosis with genes that carry disease, that are responsible for disease. So this then allows us to start to map genes involved in disease. It provides information on diagnosis, prognosis, and response to therapy, because as I say, our ability to respond to drugs is written into our genome, and we can predict drug responses now by looking at the polymorphisms expressed throughout the genome. This incidentally is not very expensive to do, and it's comprehensive and extremely informative. It also provides information on pathophysiology, and what I mean by that is the actual mechanisms of disease development, because once we identify the genes that are close to the polymorphisms, we can start to analyze their function using mouse models and really understand the molecular basis for disease. This was not possible before the genome sequence, but it's actually revolutionized the way that we can deal with disease and hopefully treat it. It's not fully lived up to promise yet, but it will do. So personalized medicine is something that has evolved from genome sequencing. Many of you who have children will be aware of Guthrie cards. Guthrie cards are what have been traditionally taken from children. A little stab in the heel and the blood spots are taken to diagnose very common ailments. They're really of very little use now. But what will happen eventually with the genome sequence is that your entire genetic barcode will register at birth. Your polymorphisms will be registered because they'll predict disease susceptibility. They'll predict response to therapy and also prognosis in terms of disease. So these polymorphisms and their analysis is going to be extremely important in personalizing medicine to you and to others as we go forward. And the transcriptomic analysis of gene expression will also contribute to this. So gene expression and single nucleotide polymorphisms will be diagnostic and predictive response to therapy. And this is the basis of personalized medicine, taking it from individual genes predicting responses to really extremely genome-wide responses. So that's all I want to say. I hope that was of some use to you. It's hard to know exactly how to pitch this, but we're going to take some questions now, and I'm sure we can take questions by email as well at the bottom of the screen. So thanks for your attention, and I apologise for talking through my nose through most of that.
Jerry, thank you very much for that. Under very difficult circumstances with the technology trying to sabotage you, you didn't falter.
No, it kind of worked.
So I've just got a couple of questions for you that I've been thinking about. Do you think that eventually we'll see all human and animal disease in terms of genetics?
Not all of it. A lot of it is driven by the environment. Many diseases, yes. The other problem with defining multigenic diseases is that the more and more genes that you define as being involved in a disease, the less their individual impact, and the less you can actually understand clearly what they contribute. So we'll never fully understand disease through genetics. We know that through twinning studies. You have to understand the environment, and actually even as we understand genetics, when it gets very complex, and it was difficult to use that information effectively. So not every disease will be understood genetically. Many will. The important thing is that I think many therapies will be genetically determined. That's quite clear, and that I think will be the major advance from this.
Okay, thank you. One of the things that Curriculum for Excellence asks teachers to do, as well as a million other things, is to enable youngsters to be scientifically literate, and that includes having a knowledge of Scotland's contribution to science. Can you outline briefly for us the ways in which scientists locally are employing some of the technologies that you've discussed for us this afternoon?
Yes, so there are many major initiatives in terms of personalised medicine going ahead. There's a personalised medicine centre in the new Glasgow Southern Hospital, which is going to really take this forward, in collaboration with big industry in Scotland as well, and small industry in Scotland. Scotland are world leaders in terms of understanding gene function. Edinburgh is a major centre for understanding gene function. So this is embedded in what we do in Scotland.
Okay, that's great, thank you. And that's interesting and nice to know.
Good.
What do you see as the skills biologists of the future, which our audience are perhaps dealing with some of those, but what would you say the skills biologists of the future will require in order to contribute to this field of study?
That's a difficult one. So I'm nominally an immunologist, but I hate being called an immunologist because I think it's too restrictive. Okay. I like to think of myself as having broader interests. That being said, we now talk about scientists knowing more and more about less and less. There's so much information around that really the idea of a broad biologist is gone. People are very specialised. What we need are people who are passionate about biology. I get paid to do a hobby. That's a special thing to be able to do. We need people who are numerate, and that is an issue that we come across all the time. And we need people who understand how to talk the language of computing and biology simultaneously. So the bioinformaticians, bioinformatics is an extremely important new branch of biology, and the ones who are very, very good at it, and they're very well sought after, are people who can speak both computing and biological language and therefore can communicate with both. That's a major challenge for the future, is to get people to occupy that area. And many computing specialists and mathematicians are now turning to biology to take their skills and use them for bioinformatics, and we could not possibly do what we do or be who we are in terms of the developments we've made without them.
Okay, thank you.